Good morning. I am Dr. Pratima Pawar, Assistant Professor, Dada Patil Mahavidyalaya, Karjat. And today we are going to see a topic from animal ecology. The course code is ZO 112 for the semester first, and the chapter we are going to deal today is population. So let's see. First, we'll see the definition. The word population is derived from the Latin word popular, which means people. It is often used to refer humans, such as human population in a city, but in the biological sense, population is an assemblage of organisms, plants, or animals which belong to a single species or several closely related species occupying a particular area. If, for example, we'll see bullfrogs in a pond, grasshoppers found in a field, or pine trees in a forest. According to Wayne Edward, 1965, a population is self-regulating system. Clark in 1954 has distinguished two types of population: monospecific with individuals of the same species, mixed or polyspecific with the individuals of several species, more than one. Characteristics of population. According to Thomas Park, a population has several characteristics or attributes which are the functions of the individual. Going with the first, that is the population density, the birth rate, the death rate, age, distribution, growth form population equilibrium, the biotic potentials and the dispersal. So here we can see a roadmap of population, how population density is increased when the birth rate increases or the death rate decreases, the age distribution, the growth forms, population equilibrium, the biotic potential and the dispersal. If there is dispersal, the population density may increase or decrease. Here we can see the population density. Population density can be defined as the number of individuals per unit area or per unit volume. The units of population density differ in different cases, as for example, the number of squirrels per square kilometer, the number of earthworms or trees per acre, and the number of diatoms per liter of water. Here we can see, according to the habitat of the organism, the measurement is changing. For example, for squirrels we are taking per square kilometer, for earthworms and trees per acre, and for the diatoms per liter. Now when the individual size in a population is highly variable, some type of biomass or live weight is measured, it is used as a unit of density. Thus the unit measurement varies for different types of population. The ways of expressing population density. Population density can be expressed in two different ways: the crude density or an ecological density. Here we can see that the crude density, the name itself tells that it is a crude method. The total number of organisms of the same species within the entire habitat, whereas ecological density is the number of individuals of the same species per unit area or volume that is used by the species. Ecological density is more accurate descriptor of the population density of a species. The measurement of population density. Population density of organisms can be measured by the following methods. The total count of the individuals of all the stages or classes. That is the offsprings, that is the young ones, the adults and the older ones. It is more practical in counting the plants than animals and provides good picture of population density. We employ several sampling methods. It consists of selecting several samples and counting the organisms per unit of the sample. For example, to determine the population density of paramecium in a dense culture, the following method is employed. First of all, we will stir the culture of paramecium thoroughly and quickly. We will withdraw 1 ml of the fluid. Count the number of paramecium in this sample. Repeat this procedure for several times. Now compile them and find out the number of individuals per cubic centimeter. The sampling method for terrestrial animals. As we know, plants cannot move from one place. But when we are talking about the terrestrial animals, they have locomotion. Hence, it is very difficult to measure the terrestrial animals. For terrestrial animals, the sampling method is worked out in different way. The area of distribution of the individuals in question is subdivided into sampling units. The circular plot, the quadrant or transect method. The size of the quadrant depends upon the size of the individual being sampled. Random samples are collected from each quadrant or sampling unit. The fractional amount of the total sample area is computed and is estimated of population density is then calculated for a particular seasonal period. So we can see that the sampling of the terrestrial animals depends upon different factors. Here we can see the circular plot method wherein a habitat can be divided into several units where circular plots can be plotted. Here we can see that a 10-meter radius circular quadrant is used, whereas 1 into 1 meter quadrant for herbs is used. In the same way, 5-meter radius circular quadrant is used. So here we can see that these quadrants can be grouped according to the type of plants we are going to measure. Quadrant. Now the quadrant method is one of the unique methods used for calculating the plants as well as the insects. Okay. Here we can see the line transect method. In the line transect method, 100 meters area or a line, hence it is called as a line transect method. Okay. In this diagram, we can see that two transects have been, which are of 2 kilometer, have been marked in a particular area and they are walked along these transects so as to detect the diversity of that place. We can see that in the line transect method, whatever organism comes over the line or along the line are measured in the line transect method. Tagging and recounting method. Using the ratio of marked and unmarked individuals, scientists have determined how many individuals are in the sample. From this, calculations are used to estimate the total population size. This method assumes that the larger the population size, the lower the percentage of tagged organisms that can be recaptured, since they will have mixed with more untagged individuals. In this figure, we can see how tagging is done. Here you can see a tag in a form of a belt. Here we can see a tag, and this is a tag which has been tagged with the fish. Using the ratio of marked and unmarked individuals, Scientists determine how many individuals are in the sample. From this, calculations are used to estimate the total population size. This method assumes that larger the population, the lower the percentage of tagged organisms that can be recaptured, since they will have mixed with more untagged individuals. For example, if we take 80 deers are captured, tagged and released in the forest, and later 100 deers are captured and 20 of them are already marked. So we can determine the population size, that is capital N, using the following equation. So here we can see the process of mark recapture method, or which is also called as the tagging method. 100 animals were captured, these are tagged, then they are released. Next time again the animals are trapped. We get the tagged plus untagged animals. We apply the formula and then calculate the population size. For example, a biologist wants to estimate the size of a population of turtles in a lake. She captures 100 turtles on her first visit to the lake and marks their back with paint. A week later, she returns to the lake and captures 100 turtles. 20 of these turtles have paint on her backs, indicating that they are recaptured animals. This example is: first, she captured, that is N, 100 animals. In her next visit, she captured K, capital K, that is again 100 turtles, whereas out of the second capture, 20 individuals, that is the small k, were recaptured animals. So here we can see that the formula goes x divided by 100 is equal to 100 divided by 20. Therefore, capital X, that is the total count, is equal to 100 into 100 divided by 20 is equal to 500. Therefore, X is equal to the total count, 20 is equal to the number of the tagged individuals, 100 is equal to the number of the tagged and untagged individuals. The formula for determining the density of population is equal to D is equal to small n, that is animals captured during the first visit, divided by A total divided by T. Hope you all have understood, and thank you.